So I'll start at 6:30. Welcome everyone to our BSGCT British Society for Gene and Cell Therapy, our patient and public engagement seminar series. In our series, we try to bring you the sort of latest advancements within the field of gene and cell therapy, try to bring it to the general public, generate interest, give more information, and also take any questions. So thank you very much for attending. Just some housekeeping stuff before we start. We will take questions during the talk. As if you can add your questions to the live chat window or the comment section. We will ask the questions to our speaker at the end of the session. You will need a YouTube account to join the channel. These messages are not private, so please include your name and affiliation in your question. And I think that's all the housekeeping. So to start, my name is Sono, and I am a postdoctoral researcher at University College London. And me, alongside Raj, Juan, and Ozgi, who are all researchers at University College London, we host these PPE seminar series. And today we are very excited to host Dr. Charley from Cardiff University School of Medicine. And to introduce her briefly, she is a researcher working with Professor Alan Parker in the Viral ImmunoTherapies and Advanced Therapeutics Laboratory at the Division of Cancer and Genetics at Cardiff University. And her current research, which is funded by Pancreatic Cancer Research Fund, it focuses on smart viruses, which are engineered to only infect and kill cancer cells, which we will hear about today. And I'm really excited to hear more about her work generating these smarter viruses. And she has also worked previously in the Vital Lab, exploring the virus-host cell interactions responsible for the rare cases of blood clots following vaccination with adenovirus-based COVID-19 vaccines. So without further delay, I will pass on to Charley.
Thank you very much. And thank you to BSGCT and also for everyone here today for joining me for some science on a Wednesday evening. So yeah, as they said, I'm Dr. Charley Lovatt-Travers. I'm a research associate in the Vital Lab here in Cardiff University. And today I'm going to be talking to you a little about how we can tame and train viruses into cancer-fighting therapies. So to start off, I want to bring you sort of right down into the macroscopic world of viruses. So viruses are a collection of non-living molecules that need a host to replicate and survive, which means that they cannot replicate without infecting a host to be able to survive. So they can survive on surfaces for a while, but they cannot replicate as they are non-living. And there are estimated to be millions of different types of virus on Earth, although as of last year, 16,123 different virus species have been described. And of these, currently 219 different types of virus are known to infect humans. And as you see here from these little cartoons, viruses can come in all shapes and sizes, but they're typically between 20 to 400 nanometers in diameter. So a nanometer is 0.00001 centimeters. As you can imagine, you can't see these by the naked eye, and actually we can't see them either with sort of the microscopes that we use in our laboratory. So as I'm sure you're all aware, viruses can have can cause real immense destruction and suffering, and there's a dark side to them, specifically viruses which are in their wild type state. I'm sure you're all familiar with COVID-19 caused by the SARS-CoV-2 virus. You can have the varicella zoster virus causes chickenpox, which can later recur as shingles in adults. There's a rabies virus, polio virus, the measles virus, Ebola, and the human immunodeficiency virus, HIV or AIDS. And there's also viruses which you may be more familiar with, such as the norovirus and flu caused by influenza. So viruses are bad news. You think they can cause all this destruction, but also viruses can potentially be generated and modified so that they can be used for good. And the reason why viruses are good at being able to be used in this way is that they have a very particular set of skills, which means that if we can properly train them and tame them, we can use them for a very broad range of therapeutic applications, such as gene therapies, cancer therapies, and vaccines. So in our lab, we like to think of viruses a bit like dogs. So dogs over hundreds of thousands of years have evolved from wild wolves. So we like to think of the wild type virus as a wolf. We can then tame the virus or tame the dog to become a viral vector. So to become a domesticated dog, which means that it's now safe to be able to put into humans, and it's very different from sort of the wild type virus that we've generated it from. We can then further train the virus to become a therapeutic vector. So in this case, you can train these dogs to become guide dogs. So you can take it from this wild-type virus, you can then make it safe for humans, and then you can actually turn it into something that could have therapeutic benefit for patients. And so a majority of viruses in the clinic at the moment are used in gene therapies. So gene therapy is where you use a virus to either fix or replace a faulty gene in patients. So a lot of these are used to treat genetic disorders which are inherited. So you can use this virus to then essentially replace the faulty gene to be able to help alleviate some of the symptoms in these conditions. One of the most common viruses used for this is an adeno-associated virus. These are very small viral vectors, around 20 nanometers in diameter, and they've been used in the clinic to treat a number of different inherited disorders such as just muscular dystrophy, haemophilia, retinal dystrophy, so the retinas in the eye can cause low vision or even blindness, spinal muscular atrophy, and AADC deficiency. And by treating patients with these viruses, you can then sort of alleviate these symptoms, and it's been shown to have real good clinical benefit in certain patients. Similarly, the herpes simplex virus has been used to treat dystrophic epidermolysis bullosa. This is a debilitating condition where the upper layer of the skin is very fragile, and this is due to mutations in the collagen gene. This herpes simplex virus is actually given topically, so directly onto the skin, and it's able to deliver a complete collagen gene to alleviate these symptoms. Lentiviruses are also used in gene therapy, so in sickle cell disease. So this is where your red blood cells have a mutation in the hemoglobin, so they are this sort of sickle cell shape, and they cannot transport oxygen as well as the healthy cells. In this instance, the blood cells are removed from the patient. The lentivirus is then used to infect these cells. It then edits the faulty gene to make it functional, and then these cells are then placed back into the patient. Now, in addition to gene therapies, viruses can also be used as vaccines, and some of you in the audience may have actually received this vaccine. So this is the COVID-19 vaccine, the ChAdOx1, which is a chimpanzee adenovirus, and that was used to during the COVID-19 pandemic. The virus would infect your cells. It would then make it produce the COVID-19 spike protein on the cells. Your immune system would come in and recognize it. So you then have a spike-specific immune response against COVID-19, so that if you do then subsequently get infected by SARS-CoV-2, you have reduced symptoms and you recover quicker. But this is all amazing uses of viruses as therapy, but in our lab, we use viruses as cancer therapies. So how do cancer therapies work? So we take our smart viruses, our targeted viruses, also known as oncolytic viruses, and these have been modified so that they cannot infect healthy cells. So when they're given to a patient, it will not go into any healthy cells, and these will remain undamaged. But it will then be retargeted to a cancer cell, and it can infect the cancer cells. Once it's infected the cancer cells, the virus will then replicate. It will overtake the cell's machinery so that it uses the cell itself to help it generate more viral progeny, because as I said, viruses are non-living. And then that results in cancer cell death and release of the virus. So you get further virus replication and further infection of your neighboring cancer cells. And you also get activation of the immune system, and you generate an anti-cancer immune response. Now, our immune system is our first line of defense against a lot of infections, but it can also be activated to fight the cancer cells. So you're using your own body to attack the cancer. And we can also further modify a virus so that it inside of it encodes in its DNA for different anti-cancer drugs. So you can further stimulate the anti-cancer immune response and essentially turn a cancer cell into a drug-making factory. So in the case of gene therapies, we like to think of the virus as a guide dog, but in our case, we like to think of our cancer target viruses as police dogs. The police dog or the oncolytic virus will only attack the criminal. It won't attack innocent standers-by. And then it will signal for the immune cells to come running in and to help it take down the cancer cell. So our lab works on the adenovirus. Now, the adenoviruses are a type of virus that cause mild or even asymptomatic infections. They have an icosahedral shape, so it's 20-sided, and they're made up of an outer protein coat known as the capsid, and they have a very large double-stranded DNA genome. And when I was talking earlier about this very particular set of skills, this very large DNA genome allows us to edit it and add in quite large sized anti-cancer drugs. And there's three sort of main proteins that make up the adenovirus. There's a penton here in green, the purple hexon, and then in red you have the fiber, so you have the fiber shaft and the fiber knob. So our lab works with human adenovirus type 5, and here it is in its wild type state, so much like the wild wolf. So as I said earlier, you have these three main proteins, and all of these bind to three main receptors in the body, which would result in a lot of off-target effects and infection of healthy cells if we gave this to a patient. So the fiber binds to CAR, which is Coxsackie and adenovirus receptor, and that's expressed throughout the body. The hexon combined coagulation factor 10, which will result in a lot of the virus being taken up into the liver. And similarly with the penton, it binds to alpha B, beta 3, and 5 integrins, which results in uptake into the spleen. So obviously we don't want to give this to patients. So we have then generated Ad5 null. And essentially, I hope Remy won't mind me saying this, it's essentially useless. So this Ad5 null, we have ablated all known binding partners of Ad5. So we've introduced three key mutations into these three key proteins, and Ad5 null cannot infect anything. So this is also great, we've now made it very safe, but it is virtually useless in this vector formation, so we have to find a way of targeting it to cancer cells. So we wanted to find a tumor-specific marker, and we found that in alpha V beta 6 integrin. So what you're looking at here is immunohistochemistry slides. So samples of normal tissue or tumor tissue are sliced very thinly till they're micrometers thick, and then they're placed on a glass slide, and then antibodies are added to stain them. In the blue here are the nuclei of individual cells, so as each one is an individual cell. And as you can see here, when we stain for alpha V beta 6, in the healthy tissue we don't see any staining, which means that our virus couldn't get in. But in our tumour tissue, you can see here in the brown, we have really strong positive staining for alpha V beta 6. So this is a really nice tumour-specific marker that we can use to direct our oncolytic virus to. So we take our Ad5 null and we retarget it to alpha V beta 6, and it's now known as Adna5 null A20. And that's because we insert a 20 amino acid peptide into the loop of the fiber. And this is from hand, foot, and mouth disease. And this retargets our virus directly to alpha V beta 6. My project works on pancreatic cancer or pancreatic ductal adenocarcinoma. And in this instance, over 80% of these tumours express this receptor for the virus on their surface. And as I mentioned previously, we can further then modify our virus so that it can then also encode for anti-cancer drugs. So obviously we've now got our vector, but we want to get it into patients and we want to be able to get it to clinical trials. So what you have to do is you first have to do preclinical data to show that it's safe in living beings. And in this case, we have to use a mouse model. So in this instance, we've taken mice and we've injected tumours into their back. And they've either been infected with the wild type adenovirus 5 or Ad5 null A20. And this encodes for a gene, which means that it will emit light under certain imaging. And I hope what you can see here in this upper layer where we're using the wild type Ad5 is that it infects everything. It's all throughout the body, and there's really strong levels of the virus throughout. And this would obviously have a lot of off-target effects in patients. However, when we treat the mice with our Ad5 null A20, you can see here that the light emission is really localized, and that's because the virus is only replicating, is only present within the tumour. And what we saw from this experiment is that when we looked at the survival of these mice, so along the bottom is their time, and on the y-axis is overall survival. You can see in these circles, when the mice are not treated at all, they succumb to their illness very quickly. However, when we look at the stars, so you look at the mice which were given alpha-120, every single one of them survived. So this shows that alpha-120 can seek out and it can destroy cancer cells. So what's great now is that thanks to a lot of work in our lab and also now alpha-120 has been licensed out to a company known as Succession Therapeutics, and they've now taken this drug to clinical trial through a lot of hard work. To be able to do this, you have to raise a lot of money, and so they've got this lovely, nice video that's going to explain this all, probably a lot nicer than I have. So I'll just do that as a nice little summary.
TROEPT, the ideal immunotherapy. Cancer immunotherapies have made great progress, but current treatments have failed to deal with the huge diversity of tumor cells. To overcome this diversity, the ideal immunotherapy should have three characteristics. It should be localized to the cancer, be broad in action to kill all tumor cell variants present, and be potent in activating the full immune system arsenal to attack the cancer. TROEPT is a unique viral technology designed to do all three by delivering a broad and potent therapeutic payload only in the cancer. To achieve cancer-localized delivery, patented mutations are introduced in the TROEPT virus, stopping its natural ability to infect healthy cells. Disabling normal cell entry mechanisms enables TROEPT to be delivered intravenously and trafficked to the cancer without being neutralized by interactions with healthy tissues, a major limitation of other viral therapies. TROEPT is further engineered to enter multiple cancer cells via a cancer-specific marker. Once in the cancer, TROEPT delivers a broad and multi-pronged attack. It replicates, breaking down some cancer cells, while simultaneously delivering therapeutic transgenes, leading to the release of one or more immune-activating drugs. Both mechanisms recruit and activate local immune cells and amplify the breadth and intensity of the response. TROEPT's highly potent and local activity gives it potential on some of the hardest to treat and most widespread forms of cancer, giving patients vastly improved prospects for long-term recovery.
So, as I said, my work is using this virus to treat the pancreatic cancer. And I think sometimes it's worth just looking at statistics and reminding ourselves of how there is such a desperate need for better therapies for this cancer. So pancreatic cancer, of which over 90% are pancreatic ductal adenocarcinoma or PDAC, is the 10th most common cancer in the UK, but it's the 5th most common cause of cancer death. And unlike most other cancers, which have shown increases in survival over the past few decades, 5-year survival and 10-year survival have remained pretty unchanged for pancreatic cancer patients. And Wales is one of the worst 5-year survivors in the world. And one of the main reasons why this is because most of the patients who present with pancreatic cancer already have either locally advanced or metastatic disease. And this means that a lot of the time they're too poorly to be able to receive active treatment. So only 1 in 10 patients will receive surgery, and only around 2 in 10 will receive chemotherapy. So there's a desperate need for more targeted therapies, which are also kinder to the patient and hopefully have less off-target side effects. Now, one of the reasons why pancreatic cancer tumours don't respond well to therapies is that they're defined as immunologically cold. And cold tumours are characterised by the exclusion of anti-tumour immune cells. So these immune cells can't physically get into contact with the tumour. Pancreatic cancer is known for having this very dense fibrous case around it known as a stroma. In addition, there's the presence of immunosuppressive immune cells. So even if immune cells do infiltrate the tumour, immunosuppressive immune cells such as regulatory T cells suppress their activity. And so together, this very sort of cold tumour microenvironment means that there's a very poor prognosis for these patients, and they do not respond well to immunotherapies. Now, immunotherapies are therapies which sort of stimulate the patient's immune response to help them fight the cancer. And immunotherapies work very well in tumours like lung cancer and melanoma because they have quite hot microenvironments, which means there's quite a lot of already immune infiltrate within them. So we want to be able to heat up the pancreatic cancer tumor microenvironment. So how do we do this with our virus? Well, we like to think of making our alpha-1 now breathe fire, so we can further train our viruses to produce anti-cancer drugs from within the tumor cells. My work has two main arms. The first is we want to generate viruses which produce chemokines. Now, chemokines are very small proteins that attract immune cells to the tumor. I like to think of them like magnets, where your tumor is now expressing a lot of your chemokine and it's pulling the immune cells into the tumor. The other arm is to use checkpoint inhibitors. So even if an immune cell comes into the tumor cells can express these checkpoints on their surface, which when they bind to the immune cells prevents them having any activity. But we can use immune checkpoint inhibitors, which have been approved for the treatment of a multitude of different cancers in the UK and across the world, which bind to these checkpoints and stop the cancer cell's ability to turn off the immune system. So you can reactivate the immune system to fight the cancer. So what I like to think of it is I have a sort of triple attack against pancreatic cancer, which is going to turn it from cold to hot. You have the alpha-1, which kills the cancer cell, but this alpha-1 is also expressing a chemokine, which is going to attract more immune cells in, and it's going to express an immune checkpoint inhibitor, which is going to activate the immune cells. So essentially you have this triple-pronged attack, much like the Powerpuff Girls. You have all three ways of attacking the cancer, and together they have this sort of combined effort to take down the cancer cell. So how do we test our viruses in the lab? So a majority of our work is done in these laminar flow hoods where you're working sort of with your arms inside and there's glass in front of you. And that's because we don't want any virus coming out towards us, but we also don't want anything from us going in towards our experiments. A majority of our work is done using cancer cell lines. So these are immortalized cell lines, which are isolated from tumor cells, and you can grow them over long periods of time. And you tend to grow them in these flasks, and they stick to the bottom layer. They grow in 2D, but we can also grow them in plates or dishes for other different experiments. And they're grown in this nice pink medium, which I think always looks like a delicious cocktail, which just has everything it needs to be able to survive. And we keep our cells in an incubator of 37 degrees, so just like the temperature of the human body. And these are just some examples of what our adherent cell lines look like when we look at them down the microscope. You see here, each individual one is a different tumor cell. So now you sort of know how we test the viruses. I thought I'd end the presentation just by showing you some data and showing you sort of what we get up to in the lab. So as I said, alpha-1 kills pancreatic cancer cells. So the virus will get into the cell, it will replicate, and it will burst the tumor cell. So what we can do is we can measure cell death. So if you look here on the y-axis, we have cell death. So 100% is obviously all the cells killed compared to untreated. And across the bottom, I have 4 different pancreatic cancer cell lines. So on the left, I have high alpha-beta-6 expression. So the alpha-1 will be able to infect these. I then have a very low and a negative cell line, which means that the alpha-1 cannot infect these cells. And as you can see here in the negative, we don't have any cell death, even at the highest virus concentration used, which is in the dark blue, 2,000 virus particles per cell. When you look at these cells which do express alpha-beta-6, our virus is able to infect them and kill them. So we know that our therapy is selective and that it can kill these cells. So, like I said, my project has 2 main arms, and I thought we'd just look at the chemokine arm today. So we want to look at, can we produce this chemokine, and can we make these cells produce this chemokine? So what we do is we take our cells, we infect it with our virus, and then post-infection at multiple different points, I take the supernatant. And the supernatant is just this media that they've been growing in, and anything they release into the media is then within the supernatant. And I then do an assay known as an ELISA to measure and quantify how much of the chemokine is actually produced. And as you can see here, if you can remember from the previous slide, Panca403 and CF-PAC1 are high alpha-beta-6 expressors. On the y-axis here, we have the picograms per milliliter of the chemokine produced. And what I can hope you can see is that after infection with alpha-1, which expresses the chemokine, we're getting strong sustained production of the chemokine. So the virus can kill the cells, but it can also make them produce this chemokine. So finally, can the chemokine attract immune cells? Does it work? And to do this, we'd have a cool little assay called the Transwell assay. So we have all our cells in a 96-well plate. This is just a side-on view. So we take our viral infected media from the previous slide and we put it into the bottom here. And then in the top, you have this transwell, which has this sort of permeable membrane along the bottom, which means cells can migrate through it. We take immune cells from blood, we isolate them, we put them in this top chamber, and we put them into the incubator. And then after 8 hours, we can then see if cells have migrated through towards the chemokine. So cells either have gone through fully through, or we can detach them if they sort of come partially through the membrane. We then add a buffer, which lyses the cells in a dye, and then we use this technique to be able to then identify how many immune cells have migrated through. And in the light green here, we have AFA-20 with no chemokine, and in the dark green, we have the AFA-20 with the chemokine. And what I hope you can see is that there's a significant increase in the migration of our immune cells compared to untreated cells. So when we infect cells with our AFA-20 with the chemokine, it produces this chemokine, and this chemokine is functional, and it increases our immune cell migration towards the chemokine. So just to summarize, my talk today. So viruses can be bad, they can cause immense damage and suffering, but in the right hands, they could be great forces for good, and they could be used for gene therapies, vaccines, and cancer therapies. Our AFA-20 virus is tumor selective and it can kill cancer cells, and we can also further train AFA-20 to deliver anti-cancer medicines to the tumor. And I just wanted to point out, this is very sort of recent news, but actually the first patient has been recruited in our clinical trial using this virus, and they were actually recruited here in Cardiff, which is very exciting. And finally, science is very much a team effort. I wouldn't be able to do anything without all the members of the lab, and particularly Alan Parker in the middle here in the crown. He does wear that quite often. And everyone else in the Vital lab. And I'd just like to say, Nadala Clow and Merry Christmas, and thank you for listening.
Thank you, Charlie, for that talk. That was really interesting. I mean, I think for me it was very interesting as well because I didn't hear much about adenoviruses. So, and I really like the analogy of the dogs. That was really cool. So I can start with some questions. I guess I had kind of two questions in one. With your therapy, how many patients are you hoping to treat? As in, would it be specific on the mutation, or would you hope to be able to kind of target range of patients? And I guess along that line, I also wanted to ask are you hoping to target like multiple different types of cancers, not just pancreatic cancer?
Yeah, so what's great about alpha beta-6 is that it's expressed in a number of different epithelial cancers, so lung, breast, gastric, head and neck. So actually the clinical trial itself is looking at a whole range of cancers. So we're actually looking at treating multiple different cancer types, which is great because it's a targeted therapy, but it also can be used sort of broadly across lots of cancers.
Okay, that's good. And the just out of interest, because with this one you're targeting the alpha-5 beta-6. So are you as a lab working towards identifying other targets as well to kind of target the adenovirus?
Yes, so a research fellow in our lab, Emily Bates, is looking at targeting gliomas. So she's got a virus which is actually targeted to a different target. So the great thing about the virus is you can edit it as much as you want, and you can really play around, sort of plug and play, and redirect it to multiple different targets. So I think it's a neat little vector.
Cool. And you kind of touched upon it, the kind of you want to stimulate the immune system as well, potentially using the sort of chemokines and the checkpoint inhibitors. Have you also considered or tried like other different type of cancer therapies that are maybe already approved to be incorporated within the sort of AB genome, I guess?
Yeah, so we have a lot of different projects in our lab. So one project is looking at bispecific antibodies. So these are antibodies which bind to the immune cell and the cancer cell and essentially sort of just like bind them together so they have a reaction. And we also have a virus which delivers a prodrug, which when a patient is treated with an antifungal drug, converts that prodrug into chemotherapy. So you have a really localized chemotherapy rather than giving it systemically.
On that note, how would you kind of administer this therapy to patients? Is there any specific route, or would it be more targeted to the cancer sites?
So the whole point of retargeting the therapy is so that it can be given intravenously and it can be given systemically, and so then it will just locate to the tumor. So it's particularly with something like pancreatic cancer, it's actually really difficult to access the tumor a lot of the time. So this would just be given intravenously.
Okay. And would you still have some off-target effects? I assume because these receptors are still expressed in other cells, or healthy cells, or
there's very minimal healthy cell expression, but the genome itself of the Ad59020 also has an additional sort of safety mechanism where it can only replicate in cancer cells because there's a certain mutation that cancer cells have that healthy cells don't.
Oh, okay, that's cool. And with the administration, like, would you expect it to work better at certain cancer stages, or you expect it to work as well throughout? I mean, it's a difficult question.
Personally don't know. That is what the clinical trial is there for. But I know a lot of other trials have shown that actually when you give it, the virus itself can also infect with metastatic disease. And so you can actually get immune responses in multiple sort of tumor sites at once. But obviously a lot of these cancers are diagnosed at a later stage. So it would be great if the virus can also be effective in this case.
And a kind of question about, I guess for you as well, because you mentioned about the work you're kind of doing in the lab now. What would be like the next step for you, like in the lab, I guess, for this therapy?
So I think there's a number of different to put into the virus, which would help, particularly pancreatic cancer. There's a number of different therapies I'd like to put in, and also I'd like to sort of target that stroma, that dense fibrous capsule, and hopefully be able to break that down and help increase therapies to the tumor.
And maybe a bit of a silly or naive question. I don't know much about the current therapies for pancreatic cancer currently. So would you also compare the efficacy to current therapy? Ideally, I mean,
yeah, ideally, really the standard of care in pancreatic cancer, if you can't have surgery, is to have chemotherapy or radiotherapy. So, and that really does only sort of increase survival by a median of a few months. So we would obviously compare it to that. But I think sort of any therapy that can improve on that would be really beneficial to patients.
Cool. I think that's most of my questions. I don't think we have any more questions in the chat. Yeah. So yeah. So with that, thank you very much for staying till 6:30 to give a talk for us. It was really interesting, and I think it was just great to know about the work that you're doing there and about adenoviruses actually. So and with that, I guess we won't have any talks before Christmas. So I do want to wish Happy Christmas to everyone, and we will have another PPE session around February time, so we will put out more advertisements around the time. And yeah, with that, I would like to thank Charley again for giving this talk for us, and yeah, have nice holidays, I guess, and then we meet you next year, 2026. Thank you.